Hi, my name is Camila, and today we're going to talk about this article called A Comparative Analysis of Molecular Biological Methods for the Detection of SARS-CoV-2 and Testing the In Vitro Infectivity of the Virus. We're going to talk about SARS-CoV-2, or mainly known as coronavirus. We have to know that that the virus is from a zoonotic origin, that it mainly affects the respiratory and gastrointestinal tracts, and that it can cause fever, cough, fatigue, shortness of breath, diarrhea, loss of smell and taste. The detection, we're gonna do it by serological test and antigen virus detection with two techniques called nested PCR test and LAMP, both in nasopharyngeal and oropharyngeal human samples. For the infectivity, we have that the analysis showed that of the 427 samples that were taken, only 42 were positive, 382 were negative, and 2 were invalid tests, with only the 10.1% being positive. The main objective of this article is to describe the application of nested PCR tests in parallel with LAMP tests for the detection of the virus in 427 samples taken between October 2020 and January 2022. For material and methods, we have first the loop-mediated isothermal amplification, or LAMP, that is used for RNA extractions and to see their subsequent reactions. This is used to get more exact information about the virus. Then we have the RNA extractions and nested one-step RT-PCR. This is used for isolating the RNA molecule and is used to amplify specific RNA sequences. This is used to know the infectivity of the virus. We also have the quantity and quality control of extracted RNA and PCR products, that it is done by an RNA calculator and gel electrophoresis. The visualization of this process can be done by an UV transilluminator. As the name says, it is used to know the quality of the extracted RNA. We also have a cultivation of the virus in cell culture, that is the preparation of the cell culture media that is used for incubation and observation of the virus. A continuación tenemos los resultados de las pruebas realizadas, donde se comparan una prueba de LAMP y una de PCR. Bueno, tenemos que en ambas pruebas el primer interno, que es el encargado de evidenciar la presencia de material genético del SARS-CoV-2, se eleva claramente, se ilumina, en este caso ilumina y en este se eleva, en el fragmento 211 BP. ¿Qué significa esto? Significa que la prueba da positiva y que sí se expresa el SARS-CoV-2. Además de eso, tenemos que tener en cuenta que se tienen en cuenta todas las pruebas, teniendo en este caso el 1 es el control, del 2 al 13 son las pruebas que han dado negativas, el 14 son las pruebas positivas y el 15 es el control positivo. En estos resultados, en la figura número 3, podemos evidenciar como a comparación de la figura anterior, no hay ninguna actividad, ya que al ver las gráficas podemos darnos cuenta de que no hay ninguna activación del primer, ni en los geles de electroforesis ni en la gráfica, dando por así como resultado negativo. Y también tenemos la figura número 5, donde se evidencia el cultivo celular. Aquí tenemos el cultivo celular recién plantado, después de 24 horas de aislamiento y después de 48 horas de aislamiento, donde claramente se ve una gran proliferación. Now we have the discussion. So here we're going to say something that some other author said, and we're going to say if the article agree or disagree with this. So we have first Arctic Y, Kalbachev et al. And they said that the lower luminescence intensity of 9 of the samples when tested with the one-step variant of the method may be due to this difference, which in the presence of less viral NC in the starting sample may lead to a more visual response. And the article agree with this. The second author we have is Sirakov et al. And he said that the primers are virus-specific, regardless of the type of sample, as the tests are designed to detect the sample virus but in different hosts. And the host species could affect the results when the extraction technique is especially developed for the subsequent reactions and is an inseparable part of the kit, such as the LAMP assay. And the article agree with this. And the third author we have is Clark J.H. et al. That says that the complications that may occur when collecting swab samples from nasopharynx may affect the test results. And the article agree with this. Okay, so last we have the conclusions. The first conclusion is that the PCR results and the LAMP results were concordant. This leading us to the conclusion that both methods can be used for the diagnosis of SARS-CoV-2, and the use of one or another depends on the circumstance and the situation. And in the second conclusion, we have that PCR and LAMP are rapid and sensitive in the detection of microbial pathogens, and this is crucial for implementing appropriate treatment strategies and controlling the outbreaks. Thanks to everyone who spent their time watching this video. I hope it's useful.